Once a year, graduate students and colleagues take a float trip down the river to exchange information and test different sampling techniques.
In addition to field work and classic research methods such as light microscopes and dissection, DNA analysis methods are critical to sea Shasta research.
Sarah Bjork, who obtained her doctorate in my laboratory, is now working for Washington Department of Fish and Wildlife. Sarah Bjork's research involved looking at the pathways of infection in fish, and she used in situ hybridization, which is a technique that uses labeled DNA to stain the parasite to track the progress of infection. She discovered the parasite enters the fish from the gills. From there, it quickly moves into the blood vessel, where the parasite replicates and begins migrating, ending up in the intestine, where the parasite population explodes and causes severe damage to this tissue.
Two other important DNA analysis methods, or molecular tools, are PCR, polymerase chain reaction, and QPCR, quantitative polymerase chain reaction. These are often used in conjunction with sentinel fish exposure.
One of my first assignments was to develop a method to detect Ceratamixa shasta in environmental water samples. Why, you may ask? Well, for many years, sentinel fish exposures have been used to detect sea shasta. These fish are obtained from hatcheries, then driven to the river and placed in cages. After 3 days, the fish are transported back to the salmon disease lab, where they are monitored for signs of disease for up to 3 months.
Some of the classic signs include a swollen abdomen because of fluid buildup, a reaction to the infection.
Also lethargy, the fish tend to be quite tired and sit on the bottom of the tanks. Often you may also see a blackened tail, and the fish tend to not eat as much. If we take an anal swab from those fish and look at that sample under the light microscope, we may see spores here, such as these characteristic kidney bean-shaped spores that are typical of sea Shasta. We may see those under the microscope, and these spores are only about 10 microns long. And if these aren't visible under a microscope, then we do a molecular test, PCR, polymerase chain reaction, that is specific for sea Shasta. PCR copies DNA, so a small amount can be amplified to a level that we can detect and analyze.
Polymerase chain reaction, or PCR, uses repeated cycles of heating and cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the primed sequences and adds nucleotides to extend the second strand. This completes the first cycle. In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
After PCR has amplified the DNA,
and here you can see the results of a gel where we have our positive control and our positive samples here. So these fish have Ceratamixa shasta, whereas these fish are not infected.
through sentinel fish exposure, laboratory examination, and PCR,
we can gather information on how many fish become infected and how severe the infection is. We can place fish in the river at different sites at different times of the year to better understand when and where the parasite is present. But as you may have gathered, this method is time-consuming, labour-intensive, and of course fish die in the process. Also, PCR cannot quantify samples. We only learn whether sea Shasta is present or absent, but not how much is in the river. So we wanted to develop a method that was faster and which would allow us to count parasites directly in the water. This involved figuring out how to collect and concentrate spores on a filter and developing a molecular test that would measure the amount of sea Shasta present. There are many different pathogens in stream water, and we wanted to target just sea Shasta. So I developed a quantitative PCR or QPCR to detect and measure specifically sea Shasta. To filter the samples, we use a vacuum pump, and this helps force the water through a filter paper. The filter paper has lots of pores in it that are only 5 microns across, so it'll catch or capture all the actinospores and mixospores that might be present in the water. And as you can see though, that also captures anything else that might be present, so you can get quite a dirty filter paper by the end of filtering that 1 litre of river water. So the method will capture anything that is greater than 5 microns in size. We then fold that filter paper multiple times and place it into a microfuge tube. Then the samples are kept frozen until we wish to extract the DNA from them. And we use kits to do that, and we extract the DNA, and then we can use that purified DNA in our either PCR or qPCR reactions. Like regular PCR, qPCR also copies DNA, but it allows us to watch the cycles of DNA replication in real time to see how much sea Shasta DNA is present. So I designed different combinations of primers and probes that matched the DNA sequence of sea Shasta, and I tried several recipes to see which chemistry worked the best. The probe is quite similar to the primers in that it's just a sequence of oligonucleotides, but it sticks to the DNA sequence in between the 2 primers. And when replication occurs, it becomes separated, and as it separates, it fluoresces, and then a computer measures that amount of fluorescence. And the more DNA you have present, the more probes attach, and the more fluorescence is released once those probes become separated. So we can measure how much DNA is present in our sample. Here it's a quantitative PCR, and we run so many samples that we load them into these 96-well plates. And the plate is placed into a quantitative PCR machine, as you can see here, and this is connected to a computer, and the data we get is electronic, and we can actually see real time the copying of the DNA during the replication process, and we can see that on the computer screen. I was very excited that I could detect just one 10-micron spore in a litre of water.
Dr. Hallett demonstrated water filtering, DNA, and qPCR analysis techniques to Klamath Falls High School students.
When we started applying this method to real river water samples, we measured as many as 100 spores per litre at some sites. Knowing these numbers will help us to better understand when and where the parasite is most abundant in the river, and also to model the parasite life cycle. qPCR detects all sea Shasta DNA, whether it be from mixospores or from actinospores. But usually we are most interested in the actinospores because these are the stages that infect fish. After years of experience, we have come to understand more about the life history of the parasite. In general, actinospores are in the water in spring and summer, while the mixospores appear in the fall and winter. In the warmer months, the polychaetes are releasing the actinospores, whereas in the colder months, the returning adult salmon are releasing the mixospores. At this time, the only way to be sure if the spore DNA measured by the qPCR is from actinospores or mixospores is to see if fish can become infected from that water. Where there is any uncertainty, our procedure is to expose sentinel fish at the same time that we collect the water sample for qPCR. The qPCR data has been very valuable. It has enabled us to look in much more detail at spatial distribution. So, because instead of having to place fish at all these sites and at all these time points, we can just collect water samples instead. So water sampling has been very versatile and generated a lot more data for us.
Grant Johnson, water quality specialist for the Karuk tribe, has worked closely with the Bartholomew Lab, collecting water samples to determine parasite concentration in the lower Klamath River.
So for instance, we can look at where the parasite is distributed in the Klamath River, and we can see that tributaries here on the right-hand side contribute very little parasite to the main stem, and we can see our hotspot of our index sites is actually here at Beaver Creek. It has the most parasite present. The water sample data has also enabled us to look at the temporal distribution of the parasite, so when it is present throughout the year. And this shows very nicely that when we look at that hotspot, our Beaver Creek site, that the parasite abundance is quite low in the cooler months, but it increases in spring. It peaks then in summer here and then decreases again. You will notice that we do have an increase in September, but we attribute this to the presence of mixospores rather than to actinospores, because this is when returning adult salmon are present in the system again.
QPCR led to an important new discovery for the Bartholomew lab. When QPCR was first tested in 2004,
we systematically placed sentinel fish, in this case susceptible out-of-basin rainbow trout, in the water, and also collected water samples. And we did this up and down the Klamath River, taking 19 paired samples from the Williamson River all the way down to Hunter Creek near the estuary. And generally, wherever we detected the parasite in water by QPCR, fish became infected. However, we were surprised that QPCR values did not directly correlate with infection severity in fish. And later, when we exposed Chinook, it was strange that at the mouth of the Williamson River and also at Beaver Creek, there were high levels of parasites in the water, but the sentinel Chinook salmon only died at Beaver Creek. Why? This led us to the hypothesis that there might be different strains of C. Shasta that infect different salmonids.